So today we're going to be doing the meiosis crossing over lab. It's more of a dry lab, not a regular lab like you would think of, but what we're going to do is look at crossing over. So the problem we're looking at this lab is what is the effect of crossing over on chromosome diversity? So a little bit easier question to look at is how similar are the offspring chromosomes to the parent chromosomes? So I want you to write your hypothesis based upon that question. So remember, we always start our hypothesis with if. What are we doing? In these cases, we're going to cross over some chromosomes and not cross over other chromosomes. Then, tell me the results that you're expecting. Tell me the results you expect for the chromosomes you cross over and the results you expect for the chromosomes that you don't cross over. So in terms of results, you're telling me, will they be similar or different from the parent chromosomes? And then your because section. For your because, your reason why you expect those results, you're going to explain to me what crossing over is. So use your meiosis notes to help explain that. So now, for your materials for your experiment, you are going to need colored paper, and you just need 4 little strips of paper. They don't need to be very large, 2 different colors, about roughly that big, 2 paper clips, and colored pencils. You can use the same colors that you use for these papers, or they can be different colors. It really doesn't matter, as long as they're 2 colors that can be distinguished. Okay. So take those 4 strips of paper, so we did that. On a scrap piece of paper, draw 4 circles in the original circle, place 1 strip of each colored paper. So it's not beautiful, but here are my 4 circles. Okay. And I'm just going to start with, this is my original parent cell. So I have 1 copy of chromosome 1 and 1 copy of chromosome 1 from the other parent. So these are homologous chromosomes. Copy this into your original cell. So really you can have a blue line and a pink line. There, you copied it. So that's your original cell. Next step. Paperclip the identical strands together, forming an X. Use the figure to the right as a guide in joining these model sister chromatids. And so I'm going to grab 2 paper clips. I'm going to paper clip these together. And paper clip these together. So there I just went through interphase and made an identical copy of each chromosome, keeping those in the original cell still. Step 4. First, assume no crossing over takes place. We're going to model meiosis 1 by moving the 2 X's into 2 different cells. So I've gone through all the steps of meiosis 1, and now these 2 are separated in these 2 cells. Then I'm going to go through meiosis 2, and I am going to separate these further. So I'm going to separate this one into 2 separate cells. I'm going to separate this one into 2 separate cells. Now what I want you to do is draw that in these circles. So 1 line in each one to show what I have right here. Now I'm going to go back to number 5. Next, repeat steps 3 and 4. So I'm going to put everything back together with the paper clips. However, assume that crossing over occurs. So I'll put these back together. And put these back together. And now this time I'm going to cross them over. Now when crossing over occurs, I want you to take a look at how you can physically cross these over. Notice I can cross these 2. If I really wanted to, I could cross these 2. I cannot cross the opposite ends. And in real life, chromosomes don't actually form an X like this. It's more of this kind of shape. And then they're kind of stringy, and so they flop around, and that's why they look more like an X. And so I can cross the sides that are closest to each other. I can't cross the sides that are furthest away from each other. So the way I'm going to do this is I'm going to rip off that piece of the chromosome. I'm going to do the same thing with the other one. And I'm going to tape these to the opposite one. So repeating step 4, I'm going to model meiosis 1 by putting these 2 X's in 2 different cells. I'm going to model meiosis 2 by pulling these apart And now I'm going to draw what I have in those 4 cells. Notice that none of these cells are identical. Each one is a little bit different. So once you've got that, you're ready to answer these questions about what just occurred. If you have any questions, don't hesitate to email or stop by office hours.